Hello friends, I am Miss Varsha Gaitonde and this video is about population improvement methods. The crops are differentiated or classified into three major types: self-pollinated, cross-pollinated and often cross-pollinated. Self-pollinated crops are rice, wheat, barley, oats, chickpea, pea, cowpea, lentil, green gram, black gram, soya bean, common bean, moth bean, linseed, sesame, kesari, sun hemp, chili, brinjal, tomato, okra, peanut, potato, etc. Among cross-pollinated crops, corn, pearl millet, rye, alfalfa, radish, cabbage, sunflower, sugar beet, castor, red clover, white clover, safflower, spinach, onion, garlic, turnip, squash, muskmelon, watermelon, cucumber, pumpkin, canaf, oil palm, carrot, coconut, papaya, sugarcane, coffee, cocoa, tea, apple, pears, peach, cherries, grapes, almonds, strawberry, pineapple, banana, cashew, pasaiva, taro, rubber, etc. Often cross-pollinated crops are sorghum, cotton, triticale, pigeon pea, tobacco. I am discussing about the pollination mechanism in different crops because the pollination system is majorly a criteria to use the population improvement methods in different types of crops. First, let us see what is population and what is population improvement. Population is nothing but is a group of plants sharing common gene pool. Population improvement includes development of broad genetic based gene pool and improving through recurrent selection methods. Population improvement means the process which aims to address diversity collection to identify new genes and gene combinations that can be introduced into crops in a natural way using conventional breeding without using genetic modification but using most modern tools for selection and analysis. Importance of population improvement: it is used to adapt itself to environmental pressure, to give high yield even in stress condition, to show resistance in unfavorable conditions, for better quality, to have stability in yield even in unfavorable conditions. Development of random mating population. Here population can be developed for different purpose. Random mating means there is no mating, no control over mating, and any female has equal chance to mate with every male individuals. Here this is for improving a single trait, for selecting several traits simultaneously, for generating fertility restorer and non-restorer or maintainer populations, for deriving hybrid parents. The development of a population involves three steps: selection of component parent, introgression of GMS gene, and random mating among parents. Different population improvement approaches helps in accumulation of desirable alleles in a population through various breeding techniques known as population improvement, and these breeding procedures are used for such work which are referred as population improvement approaches. In breeding, four approaches generally used for population improvement, that is recurrent selection, disruptive selection, diallele selective mating and biparental mating. Don't get confused with population improvement approaches and methods. Approaches are what we have mentioned here, but the methods are different, that is intrapopulation and interpopulation. We'll discuss it in the later part of this presentation. The first one is population improvement approach is disruptive selection. This is used for developing superior genotypes in a population. In this breeding approach, both extreme types for a character are selected in the segregating population and intermating is done among selected parents. Suppose we want to develop early maturing genotype. To achieve this, plants with extreme earliness and lateness are selected in F2 or later generations of a cross, and mating is done between early and late plants. Several selection and mating and selection was developed by Mather in 1953 and Thode in 58 and 60. The features of disruptive selection. Method is applicable to the genetic improvement of both self and cross-pollinated species. Disruptive selection may be considered a form of recurrent selection because this method also involves selection and intermating of superior plants in segregating population. It differs from directional selection, where in directional selection only one kind of plants are selected, for example tall or dwarf, but in disruptive selection tall plus dwarf or tall and dwarf plants are selected. That is the differentiation between disruptive and directional selection. Merits of disruptive selection. It is an effective breeding method for breaking undesirable linkages, releasing genetic variability, generating diversity, and improving adaptation of plant population. Disruptive selection is useful when wild germplasm is utilized in the breeding program. It will break repulsion phase linkage in such breeding programs. The demerits are repeated crosses have to be made for improvement of particular character. Populations of limited crosses can be handled by this method at a time. Second one is diallele selective mating. The concept was developed by Jensen in 1970. He used this system for genetic improvement of wheat. Now this system is used for genetic improvement of various autogamous crops, especially for grain crops like wheat, rice, etc. Remember, population improvement methods and approaches are mainly popular among cross-pollinated crops because of their pollination habit, but in self-pollinated crops they are less attractive. The features of diallele selective mating. It is an effective method of improving those autogamous species which are difficult to cross and have few seeds per cross. Thus, it is useful in improving small grain crops. This is an important method in self-pollinated species, thus supplements conventional breeding method in autogamous crops. It generally considers a form of recurrent selection because it involves selection and intermating in segregating generation. Both mass and recurrent selection procedures are used for handling material of this system. Thus, it permits use of conventional breeding methods as well as improvement of population by intermating. Here, superior or promising genotypes can be identified and isolated for developing new cultivars at any stage of breeding program. It also permits incorporation of new germplasm at any stage in the breeding program. In the conventional breeding program, usually two parents are included in the breeding program at a time. This system permits inclusion of multiple parents. This is useful in both short-term and long-term breeding goals because it permits use of conventional selection procedures at any stage in the breeding program as well as broadening the genetic basis of population. The end product of this system can be used as a pure line variety, a mass selected variety, or as a parental line for further lines for further use in breeding program. Merits of diallele selective mating. It helps in broadening the genetic basis by incorporating multiple parents in the program. This is very effective in breaking undesirable linkage blocks because permits intermating of selected plants in segregating generations. It fastens the genetic recombination. This system also results in creation of vast genetic variability for various economic characters. This is possible due to incorporation of multiple parents in the breeding program and intermating in segregating population. In small grain crops, this system has been reported to be very effective in delaying new cultivars. The demerits are large number of crosses have to be made to obtain sufficient quantity of crossed seeds. This is a difficult task without the use of male sterility. It involves handling of materials by mass selection as well as intermating and segregating population, which requires more labor and space. This method is less effective in improving those characters which have low heritability. This system is rarely used in crop improvement and has not become very popular so far. Third one is biparental mating. It is crossing among randomly selected plants in F2 or subsequent generation of a cross in a definite fashion. The concept was originally developed by Comstock and Robinson. Important method of concentrating favorable genes in a population. 3 or 4 cycles of biparental matings are required to achieve the desired improvement. For better understanding, it is further classified as North Carolina 1, 2 and 3. 1 is called as nested design, 2 factorial design and 3 is most appropriate among the mating designs. The merits are very effective in breaking undesirable linkages. The technique leads to creation of vast genetic variability in a population for various economic characters. The technique is applicable for genetic improvement of both self and cross-pollinated species. This is an easy and effective method of population improvement. Demerits. Large segregation population has to be grown for selection and intermating to achieve desired improvement. Repeated biparental mating has to be made for 3 to 4 generations. The technique permits handling of limited segregating population at a time. Coming to kinds of population improvement. There are 2 types, intrapopulation and interpopulation improvement. Intrapopulation improvement. The improvement within a population. The most popular method is mass selection. This is based on phenotype of an individual. As you know, in mass selection, several phenotypically superior individuals are selected. They are bulked, means mass selection is done. Then they are bulked and forwarded for the next season. Family selection. This is based on family of individual plant. In family selection only, half-sieve family selection or progeny selection, where plants within each family has one parent in common. Usually the common parent is female. This requires 2 generations per cycle since it involves progeny testing. Full-sieve selection or full-sieve progeny selection. Here plants in each family is selected by mating 2 selected plants in a pair. Remember, half-sieve means one parent will be common, and in full-sieve means both parents are known and known crosses are made. Inbred means family produced by selfing or continuous intermating or inbreeding. Modified mass selection. Doggett in 1972 has described modified mass selection with intermating male sterile and male fertile plants, selection in successive generations aimed at enhancing selection response by released parental control. In one cycle, seed is harvested from only selected male sterile plants. These seeds are bulked and sown to constitute the population for the next cycle of selection, wherein male fertile plants are selected and harvested seed from selected plants is bulked for selection of male sterile plants. The procedure is continued. The mass selection should be used in the first few cycles of selection after synthesis of a population. This makes population reasonably uniform for plant height and maturity before using more sophisticated methods of recurrent selection requiring family or progeny evaluation. S1 family or progeny selection. This is one of the most effective selection schemes for sorghum as given by Gardner in 1972. This selection requires 3 generations per cycle. Just to remind or recall your knowledge, sorghum is often cross-pollinated crop. Here, heads of male fertile plants are bagged at flowering to ensure selfing, or they can be tagged to ensure that heads from male fertile plants and not male sterile plants are harvested at maturity. Selected plants are harvested and threshed separately, each head forming the S1 family. These families are evaluated in yield trials. Remnant seeds or remaining or seeds kept for the next season from the families selected or the recipped families based on the yield trials is sown, and seed from male sterile heads are selected to ensure recombination. Seeds from male sterile heads are then bulked and sown. Male fertile heads of good plants are identified for testing to begin next cycle. The units of selection and recombination are S1 progenies. The basic concept behind self-progeny selection is to expose deleterious recessive genes to facilitate their elimination during evaluation and to increase additive genetic variation. Coming to S2 family or progeny selection. In this scheme of selection process, heads of selected male fertile plants are bagged at flowering to ensure selfing, or they can be tagged to ensure that heads from male fertile plants and not male sterile plants are harvested at maturity. The S1 progenies are grown and plants in F1 progeny rows are selected and again selfed. Selected selfed plants are harvested and threshed separately, each head forming an S2 family. These are evaluated in yield trials and handled exactly in a manner similar to that of S1 progeny selection, and thus selfed or inbred progenies constitute units of selection in both the methods. S2 progeny testing is expected to result in maximum gain per cycle and is most suitable or suited when 2 growing seasons are available per year, thus permitting 1 cycle every 2 years. The method has several advantages over the other, like additive genetic variance is maximized in S2 families. The S2 are sufficiently uniform to permit precise evaluation. 2 generations per year provide sufficient time between the generations for sending seed to test locations in a range of environments and analyzing the data for selection of lines for recombination. Selection for different traits can be done in various generations ranging from half SIP to S2 according to nature of their inheritance. And the lines evaluated are more homozygous, and it is hence easier to extract pure lines. In addition, continuous selfing and evaluation is expected to improve the probability of deriving more vigorous inbred lines. The advantage of this scheme lies in the necessity of SIB mating S2 lines to increase the frequency of male sterile plants for reconstituting the next cycle of population. Test cross family or progeny selection. This method is slight deviation from the concept of intra-population improvement in the sense that the targeted population is improved based on the evaluation of test cross progenies generated by crossing several selected plants of targeted population with a broad-based tester population. The targeted population is not only improved for per se performance but also with respect to general combining ability. The steps are: first one, self-pollination of male fertile plants and test crossing them to a broad-based tester. In the second stage, evaluation of test cross progenies for yield trials, and in the third stage, intercrossing of selected plants based on yield. Coming to interpopulation improvement. Two population improvement improved simultaneously for combining ability with each other. Improvement between the populations, and recurrent selection is mainly used in interpopulation improvement. Here, recurrent selection with half-ship family selection and recurrent selection with full-ship family selection are used. Combining ability. Before we go to recurrent selection, understanding the combining ability is very important because some of the types of recurrent selections are generally used or tested for combining ability. Combining ability is the capacity of an individual to transmit superior performance to its progeny. It is a phenomena with which inbred lines, when crossed, give rise to hybrid vigor. Progeny tests should be performed to predict combining ability of the character governed by recessive genes. Types of combining ability: general combining ability and specific combining ability. General combining ability is average performance of an individual in a particular series of crosses. The features are: it is due to additive genetic variance and additive into additive gene interaction. That means it can also be estimated by additive into additive kind of epistatic interaction. It denotes combining ability of genotypes, especially inbred with various testers. It helps in identification and selection of best genotypes to use in hybridization as a parent. It is estimated by half-ship mating. It have relationship with narrow genetic or narrow sense heritability. Specific combining ability. It is the performance of a parent under consideration in a specific cross. The features are: it represents deviation from GCA. It is due to dominance, genetic variance, and all the three types of gene interaction. It helps into identification and hence selection of best cross combinations, that is, those with the desired output. When we see that an inbred combines well in any cross, it is due to specific combining ability, which is estimated by full-ship mating and have relation with heterosis. Recurrent selection. When we were discussing about the population improvement approaches in the previous section, we just discussed about the biparental mating, diallele selective mating, and one more, but here we are discussing about recurrent selection. Recurrent selection is reselection of plants generation after generation with interbreeding of selected plants to provide better genetic recombination. The procedure was described by Jenkins in 1940. The term was given by Hull in 1945. This is efficient breeding method for increasing frequency of superior genes in a population. It helps in maintaining high genetic variability in a population due to repeated intermating of heterozygous population. Recurrent selection was first suggested by Jenkins in 1940 and named by Hull in 1945. Types are simple recurrent selection for general combining ability, specific combining ability, and reciprocal recurrent selection. The first one is simple recurrent selection. It is based on phenotypic character of plants. No tester is used in this scheme. It does not measure the combining ability, and this method is useful only for those characters which have high heritability. The breeding scheme of simple recurrent selection. One cycle of recurrent selection or simple recurrent selection takes 2 seasons or 2 years. In the first method or step, superior phenotypes are selved. Seeds are harvested in bulk. In the second year or second season, individual plant progenies are planted. All possible intercrosses are made and seeds are composited. And these composited intercrossed seeds are planted in the next season, and then selfing is done. In the fourth year, individual plant progenies are planted, all possible intercrosses are made, and seeds are composited. The cycle of 2 years are repeated continuously for the accumulation of desired genes by phenotypic selection. Second one is recurrent selection for GCA. This is based on heterozygous tester cross performance. It involves general combining ability of population for a character. This method is used for genetic improvement of quantitative character. This improves those characters governed by additive gene action. This is the scheme of recurrent selection for GCA, where a block is taken and self-pollination was done in that particular block, and to the same plant population a common tester is taken and crossed. In the next step, a replicated yield trial of the crossed seeds are conducted and self-pollinated seeds are forwarded to the third year, whereas the replicated yield trial of crossed seeds are taken in the second year. In the third year, the better or good seeds of which performed well in the replicated yield trial, means the selfed seeds of better performed cross seeds are selected in the third year and intercrossed in all combinations. This completes one cycle of selection, and this will be repeated for accumulation of desirable characters. And here the tester used is heterozygous tester with a broad genetic base. Coming to recurrent selection for SCA, the main difference between recurrent selection for GCA and SCA, here narrow genetic base tester is used and homozygous tester. This is for improving specific combining ability of population and method used for genetic improvement of polygenic characters. This is the schematic diagram. There is no difference from the previous one, but the only difference is here narrow genetic base tester and homozygous tester is used. Coming to reciprocal recurrent selection, this is most important. The scheme was proposed by Comstock and co-workers in 1949. Here two heterozygous testers are used for crossing. This is to improve both the general combining ability and specific combining ability of a population. This method is used for improving polygenic character, and this is known as recurrent selection or recurrent reciprocal half-sheaf selection. This is how the schematic representation runs. Two populations are taken at a time. Population A acts as a tester for population B and vice versa. What we'll do in each population, will go for selfing, and the same selfed plants are also used for crossing, means some pollen is used for selfing and some pollen for crossing purpose. In the second year, what we'll do, we'll go for the test cross of these, means we'll go for the replicated trials of those seeds which we did cross pollination, means we just used for tester and the different populations. And what we'll do in the third year, the selfed plants which performed better in the test cross, means where test cross is done for those seeds used in crossing. Okay, the parents of those cross plants which perform better in test cross, that selfed seeds of those cross pollinated seeds are taken and they are planted in the third year, and they will be planted in separate blocks. These composite seeds of intercross are raised, and this completes the first recurrent cycle, and that can be repeated for the better identification or accumulation of desirable characters. Here you have to remember one thing: in simple recurrent selection you only need two generations, whereas in recurrent selection or for GCA or SCA or reciprocal recurrent selection, we need 3 years or 3 seasons to complete one cycle. Among the four, reciprocal recurrent selection is better because here we can go for both GCA as well as HCA, and two populations can be developed and improved simultaneously. Coming to merits of recurrent selection. This is an efficient breeding method for increasing the frequency of superior genes in a population. It helps in maintaining high genetic variability in a population due to repeated intermating of heterozygous population. The selection is made on the basis of test cross performance, and only selected plants are allowed for intermating. Demerits. It involves lot of selection, crossing and selfing work. This method permits selfing, which leads to loss of genetic variability. The method is not used directly for the development of new varieties. Coming to synthetic variety. There are two kinds of populations, mainly synthetics and composites. This synthetics is a variety developed by selecting a number of inbreds with good general combining ability. Here, intercrossing them in all possible combinations and mixing the seeds of all the crosses conducted in equal quantity. Features of synthetics. This is heterogeneous. Variety can be developed by using clones, inbreds or open pollinated varieties. Cross-pollination is must here, maintained by open pollination. Unlike composites, the exact reconstitution of synthetics is possible. This is more adaptive to varying growing conditions as compared to hybrids, less uniform than hybrids. Less attractive than hybrids, some amount of heterosis as compared to open pollinated variety have better disease resistance. Constituent genotypes can be 2 to 10. The number means a lot to achieve maximum heterosis. Heterosis increases with the increase in number of contributing genotypes, but only up to 6 genotypes. Decrease when number increase up to 10. It also depends on the crop kind which is used in developing the population. In case of forage crops, it may vary up to 50. Farmer can use his own saved seeds for 4 years, as there is no reduction in yield up to that period. After that, seed should be replaced. Low yielder than single cross hybrids or double cross hybrids, but high yield higher than inbreds. Production is less costly as compared to hybrid varieties. Production method. Varieties can be developed from well-established and tested inbred lines from S0 or S1 plants. Development or production of synthetic variety from established inbred lines. The procedure is: inbreds are selected at the starting. Those are crossed with common tester or top crossing to test combining ability. Seeds from the crosses are harvested separately. The crossed yield trials to evaluate these top crosses with checks are done. Identification of inbreds with good general combining ability. All possible single crosses among selected inbred lines are made and seeds are harvested separately. Seeds from single crosses are mixed in equal quantity. Seed multiplication by open pollination with isolation for 1 or 2 generations, then go for varietal release. In clonal propagated crops, the selection or selected base clones are kept to form new synthetic one seed regularly. Then polycross selected superior clones. From there the cultivation is propagated until synthetic 2 to synthetic 5 to obtain enough seeds. Here, clone with high combining ability. With well-combined clones, the synthetics can be more uniform and vigorous than traditional open-pollinated cultivars. Intercrossing to form synthetic 1 and open pollination to form synthetic 2 is most important step in clonally propagated crops. Production of synthetic variety from plants. First, isolation of one generation self-fed lines, evaluation of those lines for GCA, also their testing for yield and other characters with top crossing, then intermating the inbreds with good GCA either by single cross or top cross or polycross, then seeds harvested separately, finally seed multiplication and distribution. Example, maize breeding program at CIMIT Mexico and pearl millet at ICRIS at Hyderabad are based on synthetic varieties generally developed through population improvement. First one is Bajra ICMS 7703 and coconut PCA Sin variety 001. Merits. It offers a feasible means of utilizing heterosis in crop species where pollination control is difficult. Farmer can use the grain produced from a synthetic variety as seed is raised to next crop. In variable environments, synthetics are likely to do better than hybrid varieties. Cost of seed in the case of synthetic variety is relatively lower than that of hybrid variety. Seed production of hybrid varieties is more skilled operation than that of synthetic varieties. These varieties are good reservoirs of genetic variability. The good evidence that performance of synthetic variety can be considerably improved through population improvement without appreciably reducing variability. Demerits. The performance is usually lower than that of the single or double cross hybrid because synthetics exploit more GCA while hybrid varieties exploit both GCA and SCA. The performance of synthetics is adversely affected by lines with relatively poor GCA. Such lines often have to be included to increase the number of parental lines making up the synthetic, as lines with outstanding GCA are limited in number. Synthetics can be produced and maintained only in cross-pollinated crop species, while hybrid varieties can be produced both in self and cross plants. Coming to composite variety production. Selection of parental material from diverse lines, intercrossing the selected parental genotypes, evaluation of the crosses, superior heterotic combinations or progenies with desirable heterosis are selected and advanced to further generation. Parental seeds of superior crosses are mixed to constitute the variety and release of variety. Before going for any selection process in the last stage, checking for inbreeding depression and not going for testing the GCA are two important steps in composite variety production. When the plants or lines from F2 show no inbreeding depression, such lines can be identified, composited and used to release a variety. Example in maize, Ambar, Sona, Vikram, Shanti, Jawahar, Shakti, Vijay, Kisan, African Tal, Panchganga. In Bajra, MH179, WCC 75, HB3. Composite 1 has been evolved in Brassica campestis variety Toria. Was developed by compositing 10 elite Torial strains. It matures in 100 days, exhibit profuse branching and yield of about 11 quintals per hectare seed, which contains about 40% oil. Coming to the major differences between synthetics and composites. In synthetics, inbred lines should be 6 to 8 or less or somewhat more than that, but in composites it is more or less than 20. In synthetics, GCA are tested, in composite not tested. Synthetics performance can be predicted. It is broad based. Reconstitution is easy. The seed replacement is after 4 to 5 days, whereas in composites this is more broad based, cannot be reconstituted, and the replacement is after 3 to 4 years. Thank you. I hope you liked my video and kindly subscribe to get the further updates.